Hello dear students, welcome back again to Chinmoy's YouTube channel. So I hope you have already gone through the parts of photosynthesis which I have already taught. So I have already taught you what is photosynthesis, the nature of the process itself. I have taught you the importance of photosynthesis, the internal structure of the chloroplast, the opening and the closing of the stomata, and I have already taught you the equation, that is the chemical equation of photosynthesis. I have taught you the light phase, that is the photochemical phase, and the biosynthetic for the dark phase. I have already introduced that in your mechanism of photosynthesis, right? So I have also taught you the photolysis of water. So all the topics which are there in your curriculum I have already taught you, and adaptations in the plant for the photosynthesis has already been taught in the previous classes. So please go through the episodes which I have already taught. I will put the link in the description box as well as in the I link above. So let us start with today's episode of experiments to show the necessity of light, carbon dioxide and chlorophyll, and also the formation of starch and the output of oxygen. So let us start with today's episode. So what are the learning objectives of today's lesson? So in today's lesson we will be studying about what is destarching. So to test a leaf for the presence of starch, to show chlorophyll is necessary for the photosynthesis, sunlight is necessary for photosynthesis, carbon dioxide is necessary for photosynthesis. So these are the important factors which are necessary for photosynthesis, and we are just proving it with the help of experiments, and we will show that oxygen is given out during photosynthesis. So this carbon cycle will be remaining for the next class. Okay, so this I'll be teaching in the next class, and this is also a very important topic, carbon cycle. So for that you need to subscribe to my channel so that you can keep on getting updates which I am posting regularly while I am teaching in regular classes. So till today's lesson we will be learning about all the important experiments of photosynthesis. So let us start with today's experiment, and stay tuned, stay updated, and don't forget to subscribe to my channel. Now let us first discuss what is destarching. So as we all know that the presence of starch is regarded as the evidence of photosynthesis. So what does that mean? So if photosynthesis has happened in a plant, so there will be starch already present in a plant. So the experimental plant should not have starch in it, should not have starch in the leaves before the experiment is started. So what we can do? So if we want the leaves to be devoid of starch, that is, they should be destarched. We want the leaves to be destarched or devoid of starch by leaving it in dark place for 2 to 3 days. So we are leaving the plant in dark place for 2 to 3 days, and it is being devoid of starch. So now what we can do to conduct experiments on plants in open air, what we will do, the selected leaves must be destarched. So we can't put the whole plant inside for 2 to 3 days. We just can keep selected leaves covered by wrapping them under aluminium foil for 2 to 3 days, and then we can do the experiment upon those wrapped aluminium foil. We can remove that and we can take those leaves and we can do the experiments on that only. Okay, to conduct experiments on plants in open, so we must select a few leaves and we must wrap them in aluminium foil for 2 or 3 days, and then with the experimental setup is done upon those selected leaves. So let us first move on to the experiment. Now we can see here it is an experiment where it is a test for the presence of starch. So what this experiment is telling you, it is testing the leaf for the presence of starch. Okay, so here we can see the experimental setup. So what it is there, in the first setup we can see a beaker with boiling water. So what is being done, we are detaching a fresh leaf. A fresh leaf is being detached from plant and it is dipped in boiling water. So this boiling water, it is kept for 1 minute. So what is happening after keeping this in boiling water? It is killing the enzymes in it. In high temperature the enzymes will get destroyed and the protoplasm is being killed. So no further chemical change in these leaves takes place. So boiling will also make the cell more permeable to water. So the cells are becoming more permeable to water. Now we are boiling this leaf only with a methylated spirit in a water tub. So ethanol, with this ethanol in water bath. So this is a water bath where with methanol in this boiling water, again a beaker is inserted, in that ethanol is there. Okay, and that leaf is put inside, and it is again in water bath, again it is boiled till it becomes colorless. So till the leaf becomes colorless. For the removal of chlorophyll, it is boiled in ethanol. Is it clear to all? Okay, so after boiling, the leaf has become now brittle. So what we will do now, this hard and brittle leaf is again after decoloration, after removal of chlorophyll, is again now dipped in boiling water to make it soft again. So what is happening? It is made soft again. So now the leaf is spread flat and it is kept on a white surface such as a petri dish or a glazed tile. Here we can see that it is kept on a glazed tile and few drops of iodine solution are being poured on it. Okay, so few drops of iodine solution are being poured on it. So what is the observation? We will observe that if any part of the leaf was having starch, it would have been blue-black in color. So because in presence of iodine, starch becomes blue-black. Okay, in presence of iodine, starch will change its color to blue-black. Is it clear? So, but here what is happening, as no starch is present there, as no chlorophyll was there and the leaf was having no starch, it shows brown coloration. So what is it showing? The leaf is showing brown coloration. So the observation is that the leaf is showing brown coloration. Why? Because we are removing the chlorophyll from that and no presence of starch is there. So no blue-black color and the leaf is without starch. So the presence of starch is not there. We have proved that removal of starch by boiling it in ethanol, removal of chlorophyll, and the starch test is done with the iodine solution. So it's a very important test, and please learn this all diagrams, label diagrams with proper instructions how it has been given there in diagram, and try to write this experiment on your own, like requirements, aim of the experiment, procedure and observation. Write it into 3-4 breaks. You just write the materials required for the experiment, the aim of the experiment, the procedure and the observation. Now we move on to the next one. Now here in this experiment we are going to show that chlorophyll is necessary for photosynthesis. Now for that, what we are going to take here, we are taking a plant with variegated leaves. So what does this variegated leaves means? So these leaves means that they are having chlorophyll only in patches. You can see that chlorophyll is present only in patches. It's not full leaf is not green in color. So it is present only in patches. Now generally these leaves are found in croton plant, or we can say that this croton plants, they are having variegated leaves. There are other plants also, but you just remember a easy name, that is croton. So how we are destarching the plant? First process is we should destarch the plant leaf. So we are destarching it by placing it in 2 to 3 days in dark. Okay, 2 to 3 days we are keeping the plant in dark. So it is helping it to get destarched, as we have told in the previous one also. What is destarching? It is getting destarched. Then we are placing the plant again in sunlight or daylight for 2 to 3 hours. Again we are placing the sun under sunlight for 2 to 3 days. So then we are detaching the leaf and we are drawing its outline in a sheet of paper. Now then this test, we are testing the leaf for the presence of starch. Now see, we are testing this leaf for the presence of starch. How? We will see that only the parts which were green in color, you can see the parts which were green in color only, they were taking the were turning blue-black when they were kept in iodine solution, or when iodine solution was given on these full leaf. Only the parts which were having chlorophyll, that were having blue-black color, that is presence of starch in those regions which were having chlorophyll, and which means that chlorophyll is always necessary for photosynthesis. Okay, so this is a variegated leaf after starch test. So the area affected by starch is the blue-black region and area unaffected by starch is the colorless region. So it's clear to all of you, right? So this chlorophyll part only, it is after placing it under the sun for 2-3 days. Again, after destarching it, we have put that in a sun. So again it has regained the starch. So photosynthesis has happened in that part which had chlorophyll in it, and the parts which didn't have chlorophyll in it, they couldn't form starch. So hence it is proved with the experiment that chlorophyll is necessary for photosynthesis. Now we will come to the next one, that sunlight is necessary for photosynthesis. Now in this experiment we will show that sunlight is necessary for photosynthesis. Now what we are going to do, first we are taking the plant, we are taking the leaves of the plants, or we are taking the full potted plant and putting it in dark for 2 to 3 days. So if we are putting it in dark for 2 to 3 days, it is destarched. Okay, destarching is done. Now again we are taking out the plant under sunlight, but one leaf, what we are doing with a black paper, we are covering that one part of the leaf with black paper. We are fully clipping it with black paper, but the leaf is attached to the plant only. Now after 2 to 3 hours we are taking that out leaf, we are taking that leaf particular which was covered, or which we have covered one part of the leaf clipped with paper, black paper, so that sunlight shouldn't reach that part. So it was covered in such a proper way that both the upper and the lower part was covered with black paper, so that no sunlight could reach that part. So we are taking out that plant in sunlight and keeping it for 2 to 3 hours after it was being destarched. So what we will see, we will now pluck the leaf. So we have plucked up the leaf and we have done the iodine test here. So what we have seen, the part which was covered by the black paper under sunlight, so sunlight didn't reach that part. So in this part no starch formation was seen, and in the part which was exposed to sunlight, we can see that blue black color in that affected part, that part is there. So this what proves by this experiment, that the part which didn't receive sunlight didn't have photosynthesis. So absence of sunlight is a limiting factor for photosynthesis. So if sunlight is not present, photosynthesis is not going to happen. So it's an important factor for photosynthesis. So the parts which were exposed to sunlight, they had photosynthesis, whereas the part which was not having sunlight, they didn't produce starch, and so it was this didn't change its color when iodine test was done. So hence it is proved that sunlight is also necessary for photosynthesis. So same with this experiment also, we are destarching it first, then one leaf we are covering it with black paper, and we are keeping that leaf in the sunlight. But after doing this, after 2, 3, 4 hours, in the evening when we are plucking that leaf and we are doing that color test, same thing, the sunlight, the part which didn't have sunlight didn't react to the iodine reaction, because iodine test, because no starch was present, whereas the plant leaf which was exposed to sunlight had photosynthesis. So I think it is clear to you all now, because I have explained it twice. So let us move on to the next experiment. To show that oxygen is given out during photosynthesis, this is also another important experiment. So we are showing here that oxygen is given out during photosynthesis. Now we are taking some hydrilla plant here. What is the setup, experimental setup? One hydrilla plant is taken and we are placing them inside a beaker containing water. Is it clear? Now it is covering, we are covering this with a short stemmed inverted funnel. So this is a short stemmed inverted funnel. You can see that it is a short stemmed inverted funnel, and we are slightly raising the level of the funnel so that the water should enter this funnel in a free, like it should be inserting the test tube in such a way that we can say that the free circulation of water should happen. So it is just funnel is placed just a little bit above the beaker, so that free flow of water or circulation of water should go inside the funnel. Now we are inserting a tube over the stem of the funnel. So a glass tube is being inverted, test tube is being inverted over the stem of the funnel. Now we are placing this apparatus in sunlight or bright light for 2 to 3 hours. So what is the observation? You can see the observation here in the diagram itself. So we are observing the bubbles of gases appear. So bubbles of gases are appear, and the cut ends of the stem, from the cut end of the stem, bubbles are arising. Okay, and this test proves that oxygen is being produced. So some gas is being produced. So till now we are not clear that this is oxygen only. So we need to prove that also. So if we are taking out the test tube like very slowly and we are inserting a glowing sphincter in it, the sphincter bursts into flame. So which gas it supports combustion? So that is oxygen. Okay. So we know that oxygen supports combustion. So oxygen supports combustion. So for that reason only, if we are inserting a glowing sphincter, it bursts into flame. So if glowing sphincter is introduced, it bursts into flames. So it's bursting into flames. So hence it is proved that oxygen is given out during photosynthesis, and photosynthesis is happening in this hydrilla plant also, and bubbles of air that are emerging out after keeping it in sunlight for 2 to 3 hours is nothing but oxygen. Okay, it's proved. Now we are moving on to the last experiment. Now here comes the last experiment, that is to show that carbon dioxide is necessary for photosynthesis. Now what we are doing here, again see that we have taken a destarched plant. Suppose this is a destarched plant we have taken. So we have taken a conical flask over here, right? And we have put some potassium hydroxide chemical into that. And what is the use of potassium hydroxide? This potassium hydroxide is actually absorbing the carbon dioxide. Now from the potted plant we have tried to insert one leaf carefully inside this conical flask, and we have throughout this cork is through a split cork, right? There is a closing in that mouth so that it is tightly closed. So it is a split cork. So this is a conical flask which is a split cork conical flask, right? So we are leaving this setup for a few hours. So 2 to 3 hours or 4 to 5 hours we are leaving it in sunlight. So after few hours we are taking out this leaf, okay, and we are taking one more leaf from the same plant. So we are taking this leaf, suppose we are taking this leaf also from the same plant. So we will test starch test for both. So we are going to perform starch test for both. So what is the starch test? The same test which we are doing with the iodine solution, okay. So we are doing with the iodine solution. So what we are doing with the iodine solution, how the tests are done? So we will observe the leaf in the conical flask is not turning blue black. So leaf in the conical flask will not turn blue black. Why? Because no photosynthesis has occurred in this leaf. Okay, so not turn blue black, whereas the other leaf will respond to this iodine test and it will turn blue black. Okay, so presence of starch is there in this leaf, whereas absence of starch in this leaf. So suppose this is A and this is B. So in A it will not turn blue black, so absence of starch, whereas in B presence of starch. So we are observing that. So this shows that carbon dioxide is necessary for photosynthesis, as potassium hydroxide was there, and this is absorbing the carbon dioxide inside, and no carbon dioxide was present inside the conical flask, and so no photosynthesis could happen in this leaf though chlorophyll and sunlight was present. So though two factors were present, carbon dioxide was absent, so this leaf couldn't produce starch, whereas in presence of three factors, carbon dioxide, sunlight, as well as we can say that chlorophyll, these three has led to the formation of starch, and it resulted in the formation of starch, and so it turned blue black. So hence this shows that carbon dioxide is necessary for photosynthesis. I think all these 5 experiments which I have done today is very much clear to you all. It's still if you are having any queries, any doubts, please do write in my comment box. And the next part will be carbon cycle. So the last one in photosynthesis, as I have divided this lesson into 4 to 5 parts to make it easier for you to learn each part. So subscribe to my channel, stay tuned, stay updated with all my lessons which I am posting. Thank you.